This is a paper in which they look at the relationship between the hunchback and the bicoid concentration. So these plots are some way of measuring concentration. It's relative concentration, and this is bicoid. This is hunchback. This is bicoid. So you see, it's very nonlinear. And here they are trying. They also work on this accuracy of how you could distinguish different concentrations of bicoid to have this on-off expression of hunchback. And so again, you have here the hunchback versus bicoid concentration, and then oh, that was too bad. Let me see, I don't know where I went to. Okay, so let me look at this. Anyway, so this is a measurement of the relative error with which you measure the bicoid concentration. Would be something like that along the axis, and the axis is like plotted in terms of the concentration of bicoid relative to the concentration at the mean value here. And you see that the errors in that region are almost 10%, the error with which you can determine the concentration. And so that means that the concentrations are read very accurately. And as I told you, well, this is something that I'm going to skip because you know that it's binding sites on DNA, because bicoid is a promoter for the transcription of hunchback. And so the question is how long it takes to be able to infer the concentration of the transcription factor by this binding of the sites along DNA. And these are more recent results. This is the previous experimental results were work of Gregor et al. And this is work of the group of Nathalie Dostadny in Paris, and the analysis and the theory is Alexandra Wolczak. And what they say is, well, this is what the whole picture is, and again, the nuclei, these are the nuclei. This is the region where you have the switch from on-off of hunchback, and you see that the differences in the concentration are quite small, and you should be able to detect that by the machinery of the cell. And the thing is that when you have this nuclei division, so mitosis, they reset the whole thing, and the gradient is reestablished, and the expression of hunchback is again this on-off thing, and they try to estimate how fast that was done again, and it was within 3 minutes. So pretty fast, you again have this on-off pattern of hunchback and this nice gradient of bicoid. So if you go back to the work of Berg and Purcell, as Gregor et al. Did, and say, well, what's the time that it takes to achieve a 10% error in the determination of the concentration? Well, this is sort of the scaling of the error is like that, inverse of diffusion coefficient, size of the binding site, bulk concentration, and time during which you observe. You set that to 10% and you determine the time. Well, they needed the diffusion coefficient. So they did an experiment to determine that in the same paper. And they used a technique that is called fluorescence recovery after photobleaching, which is a technique in which you have, here it was bicoid that was fluorescent. You shine a very intense laser, and you wipe out the fluorescence in a particular region. So you bleach it. And then the recovery of the fluorescence means that the fluorescent molecules that were outside the region that you bleached are going back. And from the recovery time of the fluorescence, you derive the diffusion coefficient. And they obtained a very slow diffusion coefficient. Oops. Let me see. Wow, this is, okay. Which is 0.27 micron square per second. And what I see is that, so if you use this diffusion coefficient in the region where hunchback switches from on to off, and where they could estimate also the concentration of bicoid, which in number of molecules per micron cubed is 6 molecules. This is the region that we are talking about. And so green is bicoid, blue is hunchback. And use a size of 3 nanometers. Then to achieve this 10% error, you need to average this binding thing for 340 minutes, which is much larger than 3 minutes, but it's also much larger than the time that the gradient is established. So that was a very a complicated thing. So what they said was maybe there is a spatial averaging of the messenger RNA that is produced, that is regulated by the binding of bicoid. But the thing is that this averaging in space would wipe out somehow the transition zone. So it's not the best solution. And so there are various things that we can challenge, and that's what I'm going to talk about a little bit, about this calculation. One is what is the diffusion coefficient, and there were other experiments that we interpret. And then what is the correct expression for the relative error? There have been different works and some controversy about that. I'm not going to touch that. And then the thing that this formula corresponds to what I call asymptopia, which is the observation time is much larger than the correlation times. Anyway, so let me tell you a little bit about the diffusion coefficient, because it's an interesting thing, and it's also derived from analyzing autocorrelation times. And so Alvarez et al., this is the group of Cécile Pradhan in Canada, they did another type of experiments to determine the diffusion coefficient, which is called fluorescence correlation spectroscopy. In that case, what you have is you illuminate a very small volume with your laser, and you measure the fluorescence fluctuations in those volumes, in that volume. And then from the computation of the autocorrelation function, you fit it to a curve, and you get information on how fast are the fluorescent molecules transported in and out of your observation volume. And when they did that for bicoid, for inside the embryo, so bicoid is these embryos, I forgot to tell, the way that you look at that is that you express bicoid attached to GFP. And also hunchback you can express, but here is only bicoid. And then they also look at another thing, which is this molecule NLS GFP, which is also tagged with GFP, green fluorescence protein, and it's a molecule that enters in the nuclei, but it's not bicoid. And they found different diffusion coefficients. In the case of this thing that just enters in and out of the nuclei, they found a diffusion coefficient of 20 micron squared per second. In the case of bicoid, they found many components. They could fit the decay with many components with different diffusion coefficients. And what they did was they averaged those components, and they said, well, then bicoid is diffusing at the average, so the average with the weights with which each component showed in the autocorrelation function, and so they concluded it was between 7 and 9 micron squared per second. And if you repeat the calculation, you see that you would achieve this 10% in 10-13 minutes, so it's still off with respect to what you observe experimentally. So then one thing that we worked on at some point was to understand why there was this difference of diffusion coefficients determined with the two techniques. And so let me ask you what...
you have like 15 minutes plus questions.
Okay. So the way that we interpreted these differences is that bicoid is not diffusing freely, so it's binding to other things. So what it has is what is called, let me move a little fast here, is what is called effective diffusion coefficient. So if you look at the mean square displacement as a function of time, you have, for very short times, something that scales as normal diffusion with a certain diffusion coefficient, that is what we call the free diffusion coefficient. But then when you go to long times, this scales like diffusion, but with a coefficient that takes into account the binding to slowly moving molecules. So this is an explanation of what this effective diffusion coefficient is. If you look at a molecule that moves freely, it means that it bumps onto solvent molecules, and after a certain time, it transfers a certain distance, and the square of that distance is proportional to the free diffusion coefficient times the time you observe the molecule for. Now, if the thing binds every now and then to something that is immobile there, then it stays without moving, so that in the same time it travels a shorter distance. And how shorter, it depends on for how long it remained free compared to the observation time. So basically what you see is the same kind of scaling with respect to the observation time, but with a dressed diffusion coefficient that takes into account the time it remained free divided by the total observation time. If the binding sites diffuse, then what you have is like an weighted average between the free diffusion coefficient of the molecule that is diffusing and the binding site. The thing is that these effective diffusion coefficients you can have more than one. And basically the conclusion that we arrived at by analyzing what you do in FRAP and in fluorescence correlation spectroscopy is that there are two types of effective diffusion coefficients. One that is one weighted average between the two diffusion coefficients, the free and the one of the binding sites, and another weighted average, and that one of the experimental techniques gives you one of those diffusion coefficients, and the other one can give you different components with the one that we call collective, which is this du, the one that FRAP gives you that we call single molecule, or dt, and the diffusion coefficient of the site that the thing that you're observing is binding to. And so in that way we interpreted the different experiments and said, well, because it is diffusing freely at a coefficient that is about 20 microns squared per second, but then because of the interaction with sites that are almost immobile, then it has a collective diffusion coefficient of 14 microns squared per second. So everything looked compatible, and if we again try to compute this 10% thing, we get a better estimate. It's 4 minutes or 6 minutes, but still it's not that bad, that good. And so then we started to think of the expression itself, and so I'm going to give you the last part. That we wanted to recompute the relative error. And so again, we thought of these sites S that are immobile, so that would be like the binding site on DNA. These are the free particles that come into the vicinity, which is the bicoid molecules would be. And so then we have these stochastic variables, which are how many free molecules, how many binding sites, how many free binding sites, bound binding sites you have in an observation volume. And we did all the machinery about the average, variance, relative error as before. So these are the definitions. And we need to compute the autocorrelation function of the fluctuations in the number of molecules and occupancy of the binding site. And to do that, we used something that we've been using to understand fluorescence coroscopy experiments, so with reaction diffusion equations, we linearized. Let me tell you that what we have is we can obtain some analytic approximations of the autocorrelation.